Hello, science fans. Today, we're about to embark on an incredible journey into the world of genetics. Whether you realize it or not, genetics is all around us, shaping who we are and how we look. It's one of the most fascinating branches of science, and I can't wait to explore it with you. Today, we're diving into genetics, the science of heredity. Genetics helps us understand how living things inherit traits from their parents, and how those traits are passed down through generations. It's like nature's very own instruction manual, written in a special code. Genetics explains how traits like your eye color, hair texture, or even the shape of your nose are passed from one generation to the next. Have you ever wondered why you might have curly hair like your mom, or dimples like your dad? That's genetics at work. Why do you look like your parents? It's all in your genes, tiny instructions inside every living thing. Genes are made up of DNA, and they act like blueprints, telling your body how to grow and develop. Every living thing, from the tiniest bacteria to the tallest tree, has genes. Genetics started with Gregor Mendel, a monk who studied pea plants and discovered that some traits are dominant and others are recessive. Mendel's experiments were simple, but his discoveries changed science forever. He's often called the father of genetics. His work laid the foundation for everything we know about genetics today. Thanks to Mendel, scientists can now predict how traits will be inherited and even solve mysteries about our own family trees. Over time, scientists discovered that genes are made of DNA, which is like a long twisted ladder called a double helix. The order of the DNA letters, A, T, C, and G, makes each of us unique. So how do we predict what traits offspring might have? We use a special tool that organizes all possible gene combinations from the parents. This helps us see the different ways traits can be inherited. This tool is called a Punnett Square, named after Reginald Punnett. It's a simple but powerful way to map out genetic possibilities and see what traits might show up in the next generation. It's a simple grid that helps us see the probability of inheriting certain traits. By filling in the squares, we can predict the chances of having blue eyes, brown hair, or even being able to roll your tongue. By the end of today, you'll be able to use Punnett Squares like a pro. You'll be able to solve genetic puzzles and amaze your friends and family with your new knowledge. Get ready to unlock the secrets of heredity. We're about to discover how the tiniest parts of our bodies can have the biggest impact on who we are. Put on your thinking caps. Science is cool. Let's do this and see just how awesome genetics can be. So what's a Punnett Square? If you've ever wondered how scientists predict the traits of future generations, this is the tool they use. It's a simple yet powerful way to unlock the secrets of genetics. Imagine a tic-tac-toe board, but instead of X's and O's, you're filling in the possibilities of genetic inheritance. The Punnett Square is a diagram that helps us predict the genetic outcomes of a cross between two parents. It's like a crystal ball for genes. With this tool, we can figure out the chances of an offspring having a particular genotype or genetic makeup. It's used by scientists, teachers, and students everywhere to make sense of how traits are passed down. We use it to see how alleles, those different versions of a gene, from each parent combine in their children. Each parent has 2 alleles for every trait, and the Punnett Square shows all the possible ways these can mix. The Punnett Square is based on Mendel's laws, especially the law of segregation. This law says that each parent gives just 1 allele for each trait to their offspring, and the combination determines the child's traits. To set up a Punnett Square, we write one parent's alleles across the top, and the other parent's alleles down the side. This creates a grid where we can fill in all the possible combinations. Each box inside the grid shows a possible gene combination for the offspring. By looking at these boxes, we can see the likelihood of each possible outcome. By filling in the boxes, we simulate all the ways parents' genes can mix. It's like running a mini genetic experiment on paper without needing a lab. It's a powerful way to visualize and organize genetic information, making complex ideas much easier to understand. You can even use it to predict the chances of inheriting certain traits, like eye color or dimples. The Punnett Square turns Mendel's abstract laws into something you can actually see and use. It bridges the gap between theory and real-life genetics. It's logical, simple, and incredibly useful, whether you're a student learning about genetics or a scientist studying inheritance patterns. And the best part? You can draw one in seconds, anywhere, anytime. Ready to build your own Punnett Square and predict some traits? Let's get started. Let's do our first genetic cross, a monohybrid cross looking at one trait. We'll use pea plants and focus on flower color. Purple is dominant, P. White is recessive, P. Let's cross 2 heterozygous parents, both are PP. Both have purple flowers but carry the hidden allele for white. What will their offspring look like? Draw a 2x2 Punnett Square. Each parent can give either P or P. Write P and P across the top and side. Fill in the boxes PP, Peep, Peep, and PP. PP and PP mean purple flowers. PP means white. We've completed the cross. Now, let's see what these results mean. How many purple, how many white? This is where ratios come in. Let's break it down and analyze our results. Ready for some classic Mendelian math? Let's go. Time to analyze our Punnett square results. We have 4 boxes, PP, 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 and PP. The genotypic ratio is 1 PP, 2 PEEP, 1 PP. That's 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Now let's look at the phenotypes. Any genotype with at least 1 P is purple, so PP and both PP. Only PP is white. That gives us 3 purple to 1 white. The phenotypic ratio is 3, 1. For every 4 offspring, 3 will have purple flowers, 1 will have white. This 3:1 ratio is a classic result Mendel saw in his experiments. It's the hallmark of a monohybrid cross between 2 heterozygous parents. Ratios help us summarize genetic outcomes quickly. See how simple and powerful Punnett squares are? Now let's take it up a notch. Ready for 2 traits at once? Let's tackle the dihybrid cross. Here we go. You've already mastered monohybrid crosses, where you track just 1 trait at a time. Now let's take things up a notch and try a dihybrid cross, which means we'll be following 2 traits at once. This is a key step in understanding how multiple genes can be inherited together, and it's a classic experiment in genetics. We'll use pea plants again, just like Mendel did. This time we'll focus on 2 traits: seed color and seed shape. Yellow seeds, represented by a capital Y, are dominant over green seeds, which are shown as lowercase y. For shape, round seeds, capital R, are dominant to wrinkled seeds, lowercase r. These 2 traits will help us see how genes can be inherited independently. Both parent plants in our example are heterozygous for both traits. That means their genotypes are YAR. Each has 1 dominant and 1 recessive allele for both color and shape. Each parent can produce 4 different types of gametes, or sex cells, because of the different ways the alleles can be combined. The possible gametes are YR, 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 and YR. Each gamete carries 1 allele for color and 1 for shape. To figure out all the possible combinations, we use the FOIL method. First, outside, inside, last. This helps us systematically pair up the alleles from each gene, ensuring we don't miss any possibilities. Now, set up a 4x4 Punnett square. Unlike the 2x2 square for monohybrid crosses, this one has 16 boxes, because each parent can contribute 4 types of gametes. Write the 4 gametes from one parent across the top of the square, and the 4 from the other parent down the side. This sets up all the possible pairings for the offspring. Next, fill in each box by combining the alleles from the corresponding row and column. Each box represents a possible genetic outcome for a single offspring. When you're done, each of the 16 boxes will show a different genotype combination for the 2 traits. This gives you a complete picture of all the possible genetic outcomes. It might seem like a lot of work at first, but the logic is exactly the same as before, just with more combinations to keep track of. Take your time and double-check your work. Once you've filled in the entire Punnett square, you'll be able to see every possible combination of seed color and shape that could appear in the offspring. This is where the real power of the dihybrid cross comes into play. Now let's analyze the results. Look for patterns in the genotypes and phenotypes. Which combinations are most common? Which are rare? This is how scientists uncover the rules of inheritance. Count how many offspring have yellow round, yellow wrinkled, green round, and green wrinkled seeds. Tally up each group to see the distribution. Let's count and reveal the famous ratio. You'll notice a 9, 3, 3, 1 pattern, 9 yellow round, 3 yellow wrinkled, 3 green round, and 1 green wrinkled. This ratio is a hallmark of independent assortment. This is where Mendel's next big law comes in, the law of independent assortment. It explains how genes for different traits are passed on independently of each other, as long as they're on different chromosomes. Ready to see the magic? This is the moment where all your careful work pays off, and you can actually see Mendel's principles in action. Let's break down the results. Here's what you'll find. The dihybrid cross not only predicts the traits of the next generation, but also reveals the underlying rules that govern how traits are inherited together. It's a powerful tool for understanding genetics. With your 16-box Punnett square filled, let's count the phenotypes. 9 boxes show yellow, round seeds. 3 show yellow, wrinkled. 3 show green, round. 1 shows green, wrinkled. That's the famous 9-3-3-1 ratio. This pattern led Mendel to the law of independent assortment. Alleles for different traits are inherited independently. The Punnett square makes this law visible. Traits mix in all possible combinations. Genetics is full of beautiful patterns. Ready for some pro tips? Always be clear with your notation. Pick your letters and state which is dominant. Label your Punnett square. Write parent genotypes and gametes. For dihybrid crosses, use the FOIL method to find all gamete combinations. Be methodical when counting outcomes. Go box by box. Use colors or check marks to keep track. Practice with real-life examples. Dogs, human traits, anything. The more you practice, the easier it gets. Genetics is a skill. Practice makes perfect. Keep at it. Look how far you've come. You can set up and solve monohybrid and dihybrid crosses. You know how to use Punnett squares to predict genotypes and phenotypes. You've learned the classic ratios and Mendel's laws. Now challenge yourself. Try new crosses. Make up your own problems. The more you experiment, the more intuitive it becomes. Patterns will start to jump out at you. You have the tools. Now use them. Stay curious, keep asking questions, and keep exploring. Next time, we'll use Punnett squares to predict the probability of having a male or female offspring. There's always more to discover in genetics. Until then, keep thinking like a scientist.